This video, we're going to talk about an application of vertex-weighted graphs.
The topological indices are computationally efficient, but they were not designed for macromolecules. Graphical invariants, on the other hand, are used to study arbitrarily large graphs, but are usually not computationally feasible. Previous work indicates that network science measures, based on small world properties, may be informative but does not offer local information. For possible solution, use vertex weights plus a blend of all of the above. G is the upper domination number that incorporates vertex weights. G is domination number that also incorporates vertex weights. D diameter plus radius, but these do not use the vertex weights. C the circumference does not use vertex weights. M the average degree also incorporates vertex weights. Finally, P peripheral weight also incorporates vertex weights. We clustered the amino acids using various subsets of these measures. The resulting dendrogram on the left shows how the amino acids clustered according to these measures. Our goal is to find the best measures that reflect the properties of the amino acids. We can see by the Venn diagram on the right that this set of measures does a reasonable job of capturing the properties of the amino acid residues. How can we know how well we are predicting the outcome of previously unseen experiments from our model? One way is to participate in a community challenge. We participated in one of the DREAM challenges in 2009. DREAM stands for Discussions in Reverse Engineering Assessment Methods. DREAM challenge designers aim to achieve a fair comparison of the strengths and weaknesses of the methods and clear sense of the reliability of the model that researchers create. The organizers of the DREAM Challenge are Gustavo Stolovsky, IBM Computational Biology Center; Andrea Califano, Columbia University; Robert Grohl, IBM Computational Biology Center; Julio César Rodriguez, Harvard/MIT. The sponsors of the challenge are Columbia University Center for Multiscale Analysis, Genomic and Cellular Networks; then IH Roadmap Initiative; IBM Computational Biology Center. The challenge we undertook was to predict a change in binding affinity of a protein for another protein. We were given a set of proteins that bind to another protein. Each of their corresponding amino acid sequences are shown. It is known that the amino acid sequence determines the level of binding affinity, but how we can predict that level of binding affinity is difficult. So, the challenge is based on the binding affinity for the target of each of the proteins in the top of the list. What is the predicted target if there is a mutation in the amino acid sequence? We converted each amino acid sequence that was provided to us into a numerical sequence. Each letter was replaced by a numerical sequence of length 6 using the table of values. Whereas the letters do not convey properties of the amino acids, the numerical values can convey these properties. We then used a machine learning algorithm. We trained an artificial neural network to recognize if a given numerical sequence was associated with another numerical sequence. In this way, the probability measure of binding affinity could be calculated. Our method placed second in this competition. This speaks well of the graph-based descriptors. There is reason to believe in the merit of the approach. But this was a challenge using a short part of the primary sequence. What can we now consider as an effective way to model the tertiary structure of a protein? This is a picture of a large membrane protein. It is the cystic fibrosis transmembrane conductance regulator. Mutations in the protein cause the disease called cystic fibrosis. This protein has 1,482 amino acids. The 3D structure of the entire protein has not been solved since it is so large. CFTR has 5 large domains. Nucleotide binding domain 1 is one of the 5 domains. The primary mutation that causes cystic fibrosis lies in this domain. The 3D structure of nucleotide binding domain 1 has been solved, and the coordinates can be found in the protein databank. We divided this domain of CFTR into 8 subdomains. The 8 subsequences that correspond to the 8 subdomains can be seen in the table. This domain starts with residue 488 and it ends with residue 594. We were guided, in part, by the secondary structures of the protein. In particular, we did not cover separate secondary structures when dividing the sequence into the 8 subsequences. We wrote a program in Maple that reads in a protein databank file, calculates the center of mass of each residue. Each vertex of the graph corresponds to the centers of mass of the residue structures. If 2 centers are within 8 angstroms, then the corresponding vertices are connected with an edge. In our current work, we use a Python program that Jeff wrote that accomplishes this for any protein databank file. Subdomain S2 shown below includes the most common mutation that causes cystic fibrosis. The F at position 508 is deleted in this mutation. We now investigate the effects of the mutation on our graph model. We modeled the 8 mutations. 1. Deletion of I at position 507. 2. Deletion of F at position 508. 3. Changing the G at position 542 to A. 4. Changing the S at position 549 to N. 5. Changing the S at position 549 to R. 6. Changing the S at position 549 to R. 7. Changing the G at position 551 to D. 8. Changing the R at position 560 to T. In the graph, on the right we have the subdomain before there is a mutation. On the left, notice that the vertex that represents the F at position 508 has been deleted. Thus, we have, in effect, performed our virtual mutation. We sent the mutated sequence to a free online protein fold predictor to obtain the resulting graph. Now the vertex weights for the amino acids come into play. Since each vertex in the subdomain graph represents an amino acid, we can assign weights to the vertices according to their amino acid. By incorporating the vertex weights into measure for the subdomain graphs, we can numerically represent each subdomain. These measures will change upon the mutation since the weights based on the amino acid residues will change. This change in the subdomain then results in a vertex weight in the next structure. In this way, we exploit the hierarchical nature of protein structures. Based on the weights at the top level, which differed due to mutations at the lower level, we were able to cluster the mutations in a way that reflects a role of the graph measures. In this dendrogram, we find that the mutation where G is replaced by D at position 551 is differentiated the most. However, for a different set of measures, we can change this outcome. Using a different set of descriptors, we obtain a different dendrogram. Using the principles of reverse engineering, we shall attempt to discover the structural characteristics associated with these combinatorial measures. Since our goal is to identify the drivers that cause the protein with the F508 mutation to misfold, we can now work backwards to see which graph theoretic measures separated this mutation. Information of this type is utilized in an area of research known as structure-based drug design.